Good day and welcome to our online webinar session looking at the Crystal Digital PCR, a next-generation PCR technology that enables the detection and the quantification of low-concentration targets in complex backgrounds. Let's take a look at the agenda for the presentation. We'll start off with a general introduction into Crystal Digital PCR. Then we'll follow on to assay guidelines and the design of assays for Crystal Digital PCR, followed by multiplexing strategies one can consider when using Crystal Digital PCR, then the Flex probe technology developed by Stilla Technologies, followed lastly by the hardware options available from Stilla Technologies for Crystal Digital PCR applications. If we take a look at the most commonly used applications for Crystal Digital PCR, we can see it's most commonly used as a method for absolute quantification of DNA RNA, but also for copy number variation detection, rare event detection, as well as gene expression analysis. And very often you find that this technology is very applicable in detecting low concentration copy numbers for applications like hematology or liquid biopsy assays, but also environmental testing where you're looking for a low concentration of a sample or a target in the presence of a high concentration of inhibitors or a complex background. If you look at the process of Crystal Digital PCR, we can see that it requires the same basic components as you would require for any endpoint or real-time PCR reaction. Most notably, the DNA sample, which is your backbone or target of interest that you're amplifying from. You would need gene-specific primers and probes to target a specific region of interest. Of course, the nucleotides to do the building of the new strand of DNA, the Taq polymerase to do the synthesis, and of course then the PCR tube and the buffer environment for the enzyme to be able to function correctly. And if we look at the process, it follows the very same process of denaturation at 95 degrees Celsius, annealing of the primers and probes depending on their design, but typically at between 55 to 60 degrees Celsius, annealing of the primers and probes, and then extension of those primers to form a new double-stranded DNA molecule. And this process repeats on and off for a number of times, but depending on the design of the assay, but between 35 to 40 cycles typically. And at the end, we've got an amplified DNA product. Looking at the visualization methods for qPCR as well as endpoint PCR, we can see that the first generation of endpoint PCR, we used to, after the PCR reaction is completed, load the contents of the PCR tube onto a agarose gel, run it in a tank with some buffer, and after a while you'll be able to visualize the DNA by exciting the gel with a blue-green LED or UV light. And the results would typically be what you see below. A lane loaded with a ladder that has specific bands of specific sizes, and you could determine if your band of interest is present in the gel, yes or no, and are there any contaminating bands present on the gel or non-specific amplicons present in the gel. But there was no real way to actually quantify if your gene of interest was present and how much of that gene was present. Into the new technologies such as qPCR and digital PCR, where we now have a reporter molecule that specifically targets our gene of interest, and during the amplification process will fluoresce and indicate that amplification of our target gene has amplified, and thus enabling us in this way to detect the amplification of our specific genes of interest. There are various fluorescent chemistries at our disposal to allow us to visualize DNA being amplified in digital and qPCR. The first one being the evergreen chemistry, or cybergreen chemistry as it's more commonly known. Essentially, a fluorescent molecule that inserts itself into any double-stranded DNA and starts fluorescing more intensely. It does have the drawback that the fluorescent molecule, or the cybergreen molecule, is not target gene specific. Therefore, if you have any non-specific amplification, the molecule will insert into any double-stranded DNA and fluoresce more intensely. To control for this process, we utilize hydrolyzing fluorescent probes or Taq probes as they are more commonly known, with a fluorescent reporter molecule on the one end of a nucleotide sequence that is gene-specific and a quencher molecule that is present on the other end that is responsible for absorbing the fluorescence in case these two reporters and quenchers are in close proximity. So what happens in the case when no fluorescence or no amplification takes place? This reporter molecule stays in close contact with the quencher, and when it does receive excitation from the light source, the quencher is able to absorb the fluorescence and none is seen by the instrument. But in the case of extension of your gene of interest, your primers would have annealed upstream and downstream of the pro-binding target region, and when amplification takes place, the exonuclease activity of the enzyme actually degrades this probe molecule, allowing the reporter molecule to be released from the quencher. And therefore, the distance increases between these two, and the fluorescent molecule fluoresces upon excitation with a light source. When looking at the process of quantifying an unknown nucleic acid with real-time PCR, we follow the standard curve approach. Essentially, a standard curve is a known concentration of DNA that has the target gene of interest that is diluted down over a number of dilutions, typically between 5 to 6 dilution series of a 1 in 10 dilution. This generates something similar to what you see here, called a standard curve. Each standard has a known concentration, and each concentration is associated with a CQ value. The CQ value is plotted against a curve value here, which indicates what is the CQ value versus the concentration of that DNA standard. How do we determine what the concentration of our unknown sample is? We determine what is the CQ value of that unknown sample that we are determining or trying to quantify, and plot that against the standard to determine what the concentration for that sample is. Thus, our unknown sample is quantified relative to the known standard, hence the name. If we compare the process of detecting DNA present in a sample, if we compare this to qPCR, you would see that in the qPCR bulk reaction of 20 microliters, we have a variety of wild type indicated by the green molecules and one rare mutant available in the red DNA molecule below. Thus, it is very difficult to detect this rare mutant DNA in the presence of the wild type background because of the low concentration of that sample. But digital PCR, because of the partitioning that we are seeing happening with the sample, from 20 microliter reactions, we now end up with thousands of small nanoliter sized reactions. And in these small nanoliter sized reactions, we have partitioning of the DNA molecules as well. And if you take this one droplet as an example, here we have two DNA molecules, one rare mutant type and one wild type. And you can see that in this specific one, the concentration of the rare or the mutant target molecule that we are trying to detect has now increased substantially because of the partitioning of the sample. Therein lies the power of detecting rare variants with Crystal Digital PCR. When partitioning a sample from a bulk reaction like qPCR in 20 microliters to thousands of smaller nanoliter or smaller sized reactions has some advantages. One of these being that the non-specific background or noise is being reduced, so it improves the sensitivity of the assay. The second, it allows you to absolutely quantify the target of interest, so therefore improving your precision. It also dilutes the inhibitors that might be present in your sample, decreasing the inhibition effects of those on your reaction. Let's take a look at the Crystal Digital PCR workflow. You would start off by setting up a reaction very similar to a qPCR reaction, with a bulk reaction of about 20 microliters being set up initially, containing all the components for the reaction. This would then be loaded into a chip that allows the bulk reaction to be subdivided through microfluidic channels into smaller droplets containing all the reagents necessary for amplification. On the instrument, the standard PCR cycling conditions of denaturation, annealing, and extension would then take place, with the reading and the quantification of the nucleic acids actually happening at the end point of the amplification curve. so after the reaction is finished. And essentially what we are counting here at this point is we want to look at what is the number of droplets or partitions that we have generated as part of this workflow, what is the number of positive droplets, and what is the number of negative droplets present in that sample. Following measuring the total number of droplets, the positive and the negative number of droplets, the software uses the Poisson statistics to calculate the absolute concentration of the sample. And essentially, this is a typical example of what you would see on a digital PCR run. Each of the little dots you see here on top is representing a droplet that contains the target gene of interest that has amplified to some extent. And at the bottom, you'll see a cluster of negative droplets that do not contain the target gene of interest. If we have a look at why we look at to compare Crystal Digital PCR versus qPCR, just look at the consistency of the absolute quantification that you have with the two technologies. Here we have an assay looking at the human albumin gene, and the same reaction repeated 23 times with qPCR and 23 times with Crystal Digital PCR. And below you can see we've used the standard curve method of quantification, and you can see that we relatively consistently, but with some variation, were able to quantify the concentration of the human albumin gene in those 23 repeats. But there's some variation. Compare this to the Crystal Digital PCR. Yes, there is still some variation, but it is a lot less compared to the qPCR alternative. There's a wealth of resources available from Stilla Technologies following this link here, where you can find more information and tutorials and training videos around digital PCR, how to design primers, how to design assays and optimize them, but also online statistical tools such as the Poisson law distribution, copy number variations, and limit of blank and limit of detection methods. Please feel free to make use of these should you need them in the future. Let's follow on to Crystal Digital PCR assay design. What are the considerations that we need to take into account when designing assays for Crystal Digital PCR? Typically, we use the same exact guidelines or very similar guidelines that you would for qPCR. We're looking at an amplicon size of ideally between 100 to 120 base pairs, but ideally 100 would be best. A GC content of between 40 to 60%, but if you cannot accommodate 40 to 60%, of course, you can move the boundaries a bit to 30 to 70% as well. The length of your primers should be kept to about 17 to 25 nucleotides in length, and the probe between 15 to 25 nucleotides at most. Your primer melting temperatures ideally should be between 60 to 62, 61 of course being the ideal, and then the melting temperature for the probing slightly higher at 66 to 70 degrees Celsius for these types of reactions. It's also a very nice way, and of course with all designs, you need to always check your designs and make sure that you've designed the primers and probes correctly. They are specific to your target gene of interest, but also they are not complementary to themselves or to other assays that you might want to run them with. And for this purposes, you could use the Oligo Analyzer from IDT. When pasting in your sequence into the tool, you can basically hit Analyze, and the system would then generate a report for you looking at hairpins, self-dimers, heterodimers, and also some mismatches as well. It will also give you more information on the length, the GC content, the melting temperatures, and so forth. You can see some details from those tools. This is again just a reminder of the two different chemistries we have available. Both of these chemistries work on digital PCR, although we prefer to use the Catman probe chemistries or the hydrolyzing probe chemistries on Crystal Digital PCR because they are sequence or target specific. With the process of designing and optimizing probes, you sometimes might need to shorten your probes for digital PCR assays. And in this case, you potentially could make use of NGB probes, the details of which I'm not going to go through on this slide, but it is a tool available to you to make sure that your probes are short enough so that they can have a high enough melting temperature for these assays. Another tool available in your toolbox to get probes shorter with higher annealing temperatures is to make use of locked nucleic acid bases as part of your probe design as well. When looking at the process of designing and optimizing probes, it is very important to make sure that you use the correct probe and the correct quencher, and also that your fluorophores do not overlap one another. The most common quenchers used during qPCR and digital PCR these days are the non-fluorescent quenchers, the black hole quenchers, but also camera quenchers, which are not recommended. In the design of your probes, it is recommended to keep the distance between the fluorophore and the quencher as short as possible. This is to make sure that strong quenching takes place between the reporter and the quencher, resulting in less background fluorescence or less background noise present in the sample. If you do end up with a probe that has a longer distance between the fluorophore and the quencher, you could potentially add additional quenchers or double quenched probes to that probe as well. This should reduce the autofluorescence of that probe molecule. Next, let's have a look how we evaluate the design of a digital PCR assay. It's important, especially for multiplex assays, that you always optimize them separately. So in other words, a single plex reaction before you start multiplexing. This is to ensure that you have the best amplifying conditions for each individual reaction before you put them together into a multiplex reaction. In this specific case, we can see we've got a triplex assay here, and the numbers indicated above is the separability score that is calculated by the software. The separability score gives you an indication of how well the positive population separates from the negative population. And in this case, we can see at the temperatures of 60 to 65, we have a very good separation of the first assay, probably with the best separation between them, the positive and the negative population being at 62 degrees Celsius. For the second assay, we can see the same happens, but arguably the best separation is probably around 62 to 61 degrees Celsius for the specific assay. But if we compare this to the third assay, we can actually see at between 60 to 63, there's definitely some non-specific amplification happening. But at 64, that non-specific amplification falls away. Therefore, if we're looking to add these three assays together, we need to make sure that all three of them will function equally well in the triplex reaction. Therefore, at 64 degrees Celsius, this would be the optimal reaction for the three assays as a whole. I mentioned the separability score of the reaction. So essentially, this is something that's calculated by the software for the user. It looks at the cluster of positive droplets that's present in the sample. It looks at the cluster of the negative droplets of the sample, and it has a look at the mean distance between these two clusters and associates a value to that. And this is found under the QC tab of the software. Here is another example of why verification of your design is very, very important. Here we have a simplex reaction using just probe 1, and we can see that there's good amplification, a nice cluster of single positives, and a nice cluster of single positive droplets and negative droplets below. If we have a look at the simplex of just a single reaction, we can see that there's again a nice cluster of positive droplets and a nice cluster of negative droplets down below. But when we add these as a duplex, we can actually see that the baseline or the negative droplet background is actually raised much higher compared to either of the simplex reactions. And we can look at this and see that it's essentially caused by the probe heterodimers that form between these two assays, causing the raised background levels. Another reason why it's very important to check your primers and probes in silico. You can always also adjust the primers and probe concentrations as well as you play around with the optimization of your primers and probes. You can see here are a few examples of primer concentrations at 125 nanomolar, 250 nanomolar, 375, and 500. As the concentration increases, you can see the separability between the positive and negative cluster of droplets actually increase. Therefore, higher concentrations of the probe actually in this case resulted in better separation of the specific assay. You alternatively could also look at increasing the number of PCR cycles as another method for increasing the separation between the positive and negative droplets. Here is a look at the recommended concentration of the primers and probes we need to use with Crystal Digital PCR. First, we recommend to using primers at a final concentration of 0.5 micromolars, and there are some exceptions for the probe concentrations. You can see here for the green at site 3, there's varying concentrations. So please take note of this before you design and start your Crystal Digital PCR assays. What are some of the benefits of using multiplex digital PCR? Of course, there's a big saving associated with doing Crystal Digital PCR because you're running more information and getting more information from less samples. You're using less reagents because you do not have to set up standard curves and using less space on your PCR plate. You're using less consumables and, of course, less time to set up the reaction. All of these end up adding to be savings for Crystal Digital PCR in terms of time, resources, and manpower. But you also have an increased performance. You have an increased throughput you could potentially perform, so more samples can be run in a day. You can actually do several singleplex reactions, combine them into a multiplex reaction as well. Again, saving you cost, time, and resources. Let's look at the principle of a multiplex crystal digital PCR reaction. We start off with a big bulk reaction of 20 to 25 microliters containing all the various DNA molecules and assays and reagents required for the digital PCR reaction. These are then loaded into the Crystal Digital PCR chips, which are then partitioned, and the reactions or the reagents then subdivided into smaller reactions or droplets. Following cycling of these droplets, then the reading can take place in any of the 6 different channels. And once those readings have been done, the software then converts that information into analysis graphs that we potentially could analyze to see what does the clusters of negative and positive droplets look like, and is there good separation between those. Multiplexing in Crystal Digital PCR is intrinsically a simpler process than it is in qPCR. If we take qPCR as an example, we have one large reaction that is quite complex in nature, with a lot of DNA molecules present at different concentrations. We also have a mixture of primers and probes, all present at different concentrations, and a single polymerase to do all of the amplification in this reaction. But if we take a look at digital PCR as an alternative, you can see that this process is intrinsically a lot simpler. We now have individual reaction vesicles that contain few or lower copies of DNA present in that sample. There's a less complex mixture of primers and probes present in each sample. And there's only one endpoint state that matters. Is the sample present or is it absent at the end of the day? The unique formulation of the NICA multiplex PCR mix at a 10x format offers excellent linear quantification of targets, but also consistent amplification even in triplex reactions. It is important to always evaluate your multiplex designs in simplex first. Here you can see each primer or probe of a triplex reaction first being tested in a less complex reaction mixture of a singleplex before proceeding to multiplex reactions. Other methods that are available also to adjust the reaction, digital PCR reaction performance, is looking at, for example, adjusting the primer and probe concentrations of the assay, evaluating the primer-probe interactions, as you can see here, and then also adjusting annealing temperatures and compensating for fluorescence spillover as well. Into multiplexing strategies, there are a variety of different multiplexing strategies available for digital PCR. One could design 2 assays that target 2 specific or independent genes or regions of interest, each with its own different fluorophore and different color being detected by the instrument. You could potentially also design an assay to target a SNP region or target area of interest, or you could potentially look at designing a universal reporter and then something to detect the presence or the absence of a variant, like an insertion, a deletion, or a copy number variation as well. And you see an example here of a kit that was designed by Stilla Technologies for an EGFR Crystal Digital PCR assay, looking at the 4 different exons in a combination of all of these 3 designs together. The drop-off assays, as you see down here below, are typically recommended for screening for presence of mutations, for looking for specific transcripts of interest, or alternatively for detecting pathogens present in a sample. Below is an example of the assay results obtained from this EGFR 6-color Crystal Digital PCR kit, looking at the presence or the absence of an exon 19 deletion. As you can clearly see, no droplets present in this one and some droplets present in this specific amplification plot. If we look at evergreen versus Taqman as a chemistry to quantify in digital PCR, it also becomes very important to consider when doing multiplexing. Here's an example of 2 short amplicons, amplicon B being shorter than amplicon A. If we were to use the evergreen chemistry, we would end up with a 1D plot that looks something like this. At the bottom, we have no droplets or negative droplet clusters. We would have a positive cluster droplets representing the fragments that are shorter and fluoresce less, because the evergreen binds double-stranded DNA. The shorter the fragments, the lower the fluorescence would be for that specific amplicon. And then you would have a secondary amplicon or a secondary base or a secondary population of positive droplets indicated by the positive presence of amplicon A, which is slightly longer. So you can see it's quite difficult to actually discriminate between these 2 positive populations, and it can become quite a complex situation to discriminate between. If we compare this to using the Taqman chemistry, it should be rather simple, because we have probe A, which is specific to target A, and probe B specific to target B. And here we can clearly see that there's a population containing the droplets or the amplicons for target A, and then here we've got the targets amplified from target B. Quite simple and easy to distinguish between the 2 different populations. There's also another alternative method for multiplexing in Crystal Digital PCR, and this is called amplitude multiplexing. Essentially, what it means is that we start off with one target at a lower concentration of the fluorophore that is target or gene specific. A second probe using the same fluorophore but it's target specific to another gene is entered at a higher concentration, in this example 500 nanomolars, and we do the same for target 3 and 4. But what we end up with at the end of the day when looking at the 2D plots is we see target 1 populations, target 2 populations, and then a third population containing both target 1 and 2 amplicons present in those droplets. And then the same for target 3, 4, and target 3 and 4. But the analysis is not as simple as this, because it gets slightly more complex because you get variations of all of these amplicons present combined. So this makes it slightly more tricky, and something I'd recommend you do only once you have gained a lot of experience in digital PCR assays. When we look at the systems available from Stilla Technologies for amplifying DNA, we essentially have a 3-color system, which is able to detect the blue, green, and red range of fluorophores. And then the more advanced 6-color system, which can have a look, or we utilize these specific fluorophores from FAM, YY, Atto 550 ROX, Sci5, and Atto 700, with the specific excitation and emission wavelengths given below. Stilla Technologies is also very proud to present the Neo Plus systems, and these systems have a 7-fluorophore, 7-color compatibility, which is very similar to what you have with a 6-color system, but there's a long shift dye present for the 7th color, which is also available on this instrument. If we look at the process for Crystal Digital PCR assays and the design of these assays, Stilla Technologies was really a market leader in developing these new what they call flex probes and universal reporters. Essentially, it comes down to decreasing the price of manufacturing these assays to make digital PCR more cost-effective for the everyday laboratory. Essentially, it consists of a flex probe. The flex probe is a target gene-specific region or oligonucleotide that is tagged with a smaller nucleotide that is specific to the reported gene of interest. And then a universal reporter is a oligonucleotide with a fluorophore, and only once this tagged nucleotide is released upon amplification and binds to the reporter is the fluorescence being generated. And these are the different fluorophores that are available on the NEO system, so 7 different colors in total. How does it work in the presence of a target gene of interest? Your assay with target-specific primers and probe coupled to the gene-specific oligonucleotide tag binds to its respective positions. In the presence of the target gene of interest, amplification takes place, and as synthesis happens, the exonuclease activity of the enzyme cleaves off the oligonucleotide tag and releases it to bind to the specific universal reporter that it is complementary to. And only in the presence of that specific target gene of interest will the universal reporter then generate fluorescence, which is detected by the instrument. In this method, we can have the same target specificity and sensitivity as the state-of-the-art fluorescence, but we're using the more affordable and faster to synthesize non-fluorescent flex probes to generate the fluorescence. In the absence of the target gene of interest, there will obviously be no space for the probe molecule to bind. Therefore, no amplification would take place and the release of the oligonucleotide tag cannot take place, and it is still connected to the probe. Because it is still connected to the probe, it cannot effectively bind to the universal reporter and generate fluorescence. Thus, no fluorescence is present in the sample of interest. Why would we use flex probes and universal reporters in our assay designs? Well, the answer is very simple. Because it affords us a modular approach in designing and using these assays, it gives us the ability to design flexible assays and to use the same universal reporters for multiple different assays, not to mention the cost-effective design of these probes, as well as the quicker turnaround time and the ease of assay development. Here we can see 2 different strategies that these flex probes were used for. One is in a screening assay, where we're looking at detecting the presence of multiple different mutations in the presence of a sample. In this case, V600E, V600K, V601E. Or they could alternatively be used to detect and identify specific targeted mutations, as in this case where we see BRAF V600E, KRAS, multiple different mutations. So you can use both of these assays. You can use the same reporter molecules, universal reporters, for all of these assays. In this specific slide, you can see a typical example of these types of probes being used in drop-off assays for the various BRAF, KRAS, and NRAS mutations. Here is another example of the same flex probes being used in a low-plex assay, screening for the presence of one or a few targets, but alternatively in a mid-plex assay, looking for the presence of multiple different mutations in the presence of screening for a reference gene or assay. Typically, when we have a 7-color instrument and we use one target per color, we would think that we could do a 7-plex reaction. But an alternative approach to this would be to use color combinations, where a target would be labeled with 2 colors of interest. Doing this strategy, it allows us to go to even higher plex assays and go up to 21 different targets multiplexing. Thus, the cost per reaction goes down even further using these flex probes and color combination targets. If we use color combinations in qPCR, we can see that we should be able to accurately detect color combinations if we're using one target assays. But as soon as we introduce multiple targets, it becomes very difficult to detect and distinguish which targets are amplifying in the sample. With digital PCR, however, because of the power of the partitioning, we can easily distinguish between individual droplets that have dual colored targets present in their amplification. In the event when we do see triplicate colors, for example, more than one gene being amplified in this droplet, we can essentially exclude these triple positive droplets from the analysis going forward. Alternative strategies that are also available to the color combination that we've just discussed is also the one color, one target approach, but also the amplitude multiplexing based approach, which we discussed earlier in some of the earlier slides. The question that might come up when looking at these multiple color combinations, how to interpret the data, is how do we tell the software what to look for? Here's a schematic from the software of the Stilla system, which basically highlights the process. So once the software is open, you would look at the population editor and select population editor and positivity combination view. From there, we would say we want to add a specific population. We would designate a specific color for this population, and then edit the negative population. So basically telling the instrument which colors are not associated with this population. Then we'd also define the negative droplets, in which channels do they fall, and we'd also define the positive droplets. After that, we would hit apply and then be able to use this in the analysis of any samples using these color combinations. Here's an example of a color combination assay looking to detect a 12-plex assay of 11 ESR1 mutations, all in the same reaction. And here you can see the typical output in the 2D plots of all of these 11 assays. What are the typical applications that you can use digital PCR for? We discussed these in earlier slides, but typically you're looking at cell-free or oncology-related applications. We're looking at microbiology-related applications such as wastewater testing or pathogen detection. There's apply testing and screening for plants and specific GMO testing and food sciences, not to mention forensics as well. And then, of course, the biopharma market with gene and cell therapy and genetic disorders as well. What do we need for the different components for our flex probe workflow? So essentially, we need an assay-specific primer with an assay-specific flex probe that is not attached to a fluorophore, but has a oligonucleotide specific to the universal reporters. And then, of course, we need to have either tube A or tube B, depending on which fluorophores we want to use in our assay, present as well. And then, of course, a positive control. And the NICA PCR 10 times mass mix for multiplexing purposes. If we're buying a catalog assay, what you will receive from Stilla Technologies is an assay-specific primer, as well as an assay-specific flex probe, along with a positive control for that sample. You would also just need to remember and remind yourself to buy the universal reporters as they are sold separately. What does the process look like for custom designs? So for custom assays, you receive your custom assay with your target gene-specific primers and flex probes. You can then choose which universal reporter, A or B, depending if you're using the blue, green, red, or the teal, yellow, infrared, or long shift eyes. And then with an option positive control, you can include as well. So in terms of the design process, Stilla would do the needs analysis, perform the custom design, manufacture the probes, and then wet lab test these on G blocks as well. Then followed by the custom delivery to the customer at the end. These are just the key terms and conditions of the Crystal Digital PCR assay design services offered by Stilla Technologies. In terms of platforms and reagents and consumables, what do we have on offer from Stilla Technologies? We of course have the chips, the ruby and the sapphire chip, the reagents, the different multiplex super mixers, and also the assays and kits available from Stilla. Then in terms of instrumentation, we have the NICA 6 consisting of the Geode and the Prism 6, and of course the Neo system available as hardware platforms. And also included in these systems would be the intuitive data analysis software for the interpretation of the digital PCR information. The Prism 6 is essentially the Geode, which is used for the thermal cycling and the partitioning, and in the Prism 6, which is basically used for the reading of the different fluorophores. And we can see that it includes the intuitive crystal reader software. You can analyze up to 3 chips or consumables per run, and you can use both the sapphire as well as the ruby chip and analyze up to 6 colors per chip. And down below you can see the different colors and the excitation and emission wavelengths of these colors that were used on this instrument. And we're very proud to also announce the Neo platform, which is the first of its kind 7-color automation-friendly all-in-one digital PCR system in the market. It allows you to run 768 samples per day. It allows you to even perform some automation tasks on automation robots such as TCAN or Hamilton. You can do everything in one system. It allows you the detection of 7 colors and offers you the ability to continuously load new samples and new chips as they finish on the instrument. As we mentioned, you can essentially run 768 samples per day on this instrument should you use all of the available spaces and use the sapphire chips on the instrument. Here is a schematic of what such a workday would look like. You would load the first 2 chip plates consisting of 3 chips each into the instrument, and as they are being partitioned and cycled and read, you can load subsequent plates, and as those finish, you can load more chips. So you can queue a total of up to 8 chips to run at a time on the instrument during the day. The benefit of having this new system is that each chip plate can accommodate 3 chips, and each chip can essentially be run on its own PCR program, giving you the flexibility to run multiple different assays in one day on the same system, thus giving you the throughput and the flexibility that many laboratories would require. What are the different systems that are available in the Neo range? We have the Neo E or Essential Digital PCR. We have the Neo, which is the mid-range version, and then the Neo Plus, which is the ultimate digital PCR automation platform. All of these are an all-in-one digital PCR system, which simplifies the workflow and allows you just 5 minutes hands-on time to prepare and run digital PCR assays. It all gives you the highest multiplexing capabilities of 7 specific colors, and you can also transfer assays seamlessly across these different platforms as your needs change over time. What's different between these 3 different systems? We can see that the Neo E or Essential allows you to run 1 plate per run, therefore up to 48 samples per run. The Neo runs 1 to 4 plates per run, or up to 192 samples per run. And the Neo Plus, because it has 2 thermal cyclers, can run anything from 1 to 8 plates per run, or 384 samples per run per day. We also offer the NICA IQ-OQ kits from Stilla Technologies. This is to streamline and test the performance of your instrument in a ready-to-use format. This also has defined acceptance criteria and allows you to check on an annual basis if your instrument's performance still falls within specification. Herewith, I would like to thank you for your attention, and please feel free to reach out to us at info@diagnostic.co.za for any specific questions you might have regarding digital PCR. Also, please feel free to reach out to us if you want to discuss specifics around your digital PCR projects. And please do follow us on LinkedIn. Thank you.